So, ladies and gentlemen, it's about time. Please become seated for the second session. My name is Roland Lill. I'm the spokesman of Class 2 Life Sciences. And you can imagine that we now move from complicated geology and physics to simple biology. I'm sharing this session together with Peter Schreiner, who will do the last talk, and I will do the first three presentations of biologists. Because in biology, hydrogen can be made and be consumed, and for this you need enzymes, and the enzymes are fascinating the biologists, and we have three world experts telling us various aspects of these enzymes, and also, in the end, application possibilities using biological results for making hydrogen for energy use. The first speaker is by far the farthest person, coming all the way from Bozeman, Montana, where there is an excellent metal biology center. This is probably due to the vicinity of Yellowstone Park, which is a bit away from Bozeman. Joan Broderick, she's a good friend of mine. She studied chemistry at Washington State University, then moved to Northwestern University, working with Tom O'Halloran. She did a postdoc with Joan Stubby at the MIT. And then via various stages moved to Montana, where she is a professor since 2005. And she does seminal work on what I mentioned already, an enzyme called hydrogenase, and she studies the biogenesis of it. For the awards, she was elected not only to the National Academy of Sciences, but also the American Academy of Arts and Sciences, both in 2022, and you see how other people value her work. Without further ado, I call upon Joan, and she's speaking on the use of hydrogen in the living world. Joan.
Is this one? No. Okay. No.
That one? There we go. Okay, thank you. Thanks very much, Roland, for the introduction. And it's really an honor to be here. I'm pleased to be able to tell you a little bit, or at least sort of segue us into the role of hydrogen in the living world. So first, we need to look at the question of why is there an intersection of hydrogen with the living world? Why would biology be using hydrogen? And I think part of the answer is quite obvious. This molecule H2 has a strong hydrogen-hydrogen bond. There's a lot of potential energy stored in that. And that is very useful to us. It's useful to life in general if we can harness that energy and do it in a controlled and useful way. And so that's really a statement of why life may have come to use hydrogen. It's the same reason why in the current day there's a lot of interest in using hydrogen as a green energy source. There's a lot of energy stored in that bond, and if you can harness it, you've got energy, and the waste products are purely clean because they're water. Of course, to use the energy in the H2 bond, or to make H2, catalysts are required. So if we're talking as chemists, and I'm both a chemist and a biochemist, chemistry with hydrogen generally requires catalysts, platinum, palladium, other types of rare metal catalysts that can work very well in some instances, but are rare, they're expensive, they're difficult to obtain. What you're going to see in my talk and the subsequent talks is that biology does chemistry with hydrogen using very abundant, very cheap metals like iron. And so biology really has a lot to teach us about the chemistry of hydrogen, how to make it, how to use it, and how to harness the energy. And so we have a lot to learn really from simple life forms that use and produce hydrogen. Another aspect of hydrogen that I wanted to point out, because it may not be immediately obvious until you think about it, but hydrogen is a very small hydrophobic molecule. It can easily diffuse through biological membranes, and so it's very accessible to enzymes in biology that may be intracellular, and that makes it also a very usable molecule for biology. Okay, so we've seen some pictures of Earth this morning. I have a picture of Earth as well, even though the only reason I have it here is to remind me to tell you that on the early Earth, hydrogen is believed to have been much more abundant and available for different types of reactions than it is today. We heard some talks this morning about hydrogen deep in the Earth and closer to the surface of the Earth and even coming out of the Earth. And in the early Earth, it was believed that hydrogen was much more abundant on the surface than it is today. And that presumably guided the prebiotic chemistry that was going on in the early Earth and also the emergence of life and the evolution of life on the early Earth. One of the features that were present on the early Earth were these black smokers, these very hot, energetic features with gases coming out of them that were very rich in minerals that had iron and nickel and cobalt and other types of metals present. And there's one theory of the emergence of life, that life emerged at these types of features where there was a lot of thermal energy and minerals to catalyze reactions, and also presumably plenty of hydrogen around. And so one of the things that we have thought about over the years in the work that we've done at Montana State is how minerals that may have been present in these black smokers may have played a role in the prebiotic chemistry that initially formed molecules that were precursors to the molecules of life, and that ultimately, therefore, helped support the emergence of life on the early Earth. And so what I'm showing you here are some simple iron-sulfur minerals. So the red is iron, the yellow is sulfur. This is a mineral known as McKenna-white, very abundant even now on the Earth, presumably more abundant in the early Earth. And what we know about minerals like this is that their surfaces are catalytically active for certain types of reactions. And so you could imagine, for example, small molecules like carbon monoxide and cyanide binding to the surface of a mineral, and in the presence of water, perhaps, a catalytic reaction occurring that could form some initial amino acids, which are, of course, important biological molecules. There's a lot of other interesting aspects of mineral surfaces that provide catalytic properties that were presumably important in the emergence of life. And one of these is what we call ligand-accelerated catalysis, which is the idea that when you bind something to the surface of the metal, that can actually have a compounding effect in order to catalyze reactions that are important. So among those reactions were reactions of hydrogen, presumably. And if we think about catalysts that were available on the early Earth, early minerals that are still around, things like pyrite and McKenna-white, these are iron-sulfur-based minerals. We can look at a connection between those bulk minerals and smaller mineral particles, nanoparticles even, that have iron and sulfur, or iron, nickel, and sulfur, or iron and other metals and sulfur. And those are really the key elements of catalysts that are present in a lot of other forms. For example, there's a variety of sort of simpler catalysts that can catalyze reactions with hydrogen. And then, as you're going to see today in my talk and in subsequent talks, there are these enzyme catalysts that use these same elements of iron and nickel and sulfur to catalyze the essential reactions of hydrogen that are a key to hydrogen in the living world. Okay, so in this early Earth, with all this energy and the black smokers and the abundant iron sulfur minerals, one of the earliest life forms that's thought to have emerged from all of that are the archaea. So this is just a picture of one archaea. The archaea are very abundant on Earth. They're all over the place even today. And I'm showing you some pictures from Yellowstone because this is one of the places in these very hot, hydrothermal areas, the very acidic areas where these archaea are able to use their unique metabolism, and it's a very broad-based metabolism, to live in these unusual environments. And that biochemistry of archaea that allows them to live in these kinds of places is presumably also what allowed them to be so successful in emerging and multiplying on the early Earth. So in terms of the topic of today's symposium, the reason archaea are interesting is because they encode enzymes that allow them to harvest that energy from hydrogen. Okay, so these are hydrogenase enzymes that will take H2 and convert it to protons and electrons in a controlled way to grab the energy from that bond and use it for their biochemical processes. Some archaea are also able to break down organic matter to form hydrogen, so they can really do both sides of the equation of making hydrogen and then harnessing the energy from the hydrogen during the alternate process. So archaea are important. They're important in hydrogen metabolism. They were important on the early Earth. They're also important in the modern Earth in a whole variety of ways. They're one of the very important microbes that's present in the intestines of ruminants. So I could have shown cows here, but I decided to show bison, American bison, also from Yellowstone. They're also ruminants. And in the guts of animals like this, there are archaea. They're specifically methanogens, so methane-forming archaea that use hydrogen as their food source. They will oxidize the hydrogen, and the electrons that come out of that are then used to reduce carbon. So CO2 gets reduced, ultimately forming methane. And this is the reason that ruminants, like cattle and bison and many others, emit methane, which is one of the most potent greenhouse gases on the Earth today. So hydrogen, in this sense, is also really interesting because it appears to be, or at least a very powerful hypothesis called the hydrogen hypothesis, is that hydrogen and its metabolism was really at the origin of modern eukaryotic biology. And the idea here is that archaea and bacteria had a symbiotic relationship with one forming hydrogen and the other needing the hydrogen and using it as a fuel, and that symbiotic relationship caused these two organisms to form a more complex relationship where one was inside the other, and we had the formation and the consumption of hydrogen now within a single organism within an organism. Okay, so that's the basis of a eukaryotic cell, in essence, And it's interesting to think that that really may have come about because of the central role hydrogen was playing on the early Earth when these organisms were emerging and evolving. Okay, so let's look at hydrogen today on the modern Earth, in the living world. It's a huge product of living organisms. So you can see an estimate, about 10 to the 8th tons per year are produced by life, by living organisms that are making hydrogen as part of their normal metabolism. And there's different ways that this can happen. I'm not going to go into all of the vast literature on hydrogen production in biology, but I'll give you some general categories and some of the main ways that hydrogen is produced in biology. One of these is by photoproduction. So these are organisms that harness the energy of sunlight and use that energy to produce hydrogen. Okay, so a couple of examples here: Chlamydomonas reinhardtii, cyanobacteria are both examples of organisms that can do this. Through photosystems, they take the energy of sunlight. That energy drives the transport of electrons into proteins that are called ferredoxins. These are iron-sulfur proteins, so another very ancient type of protein that has little iron-sulfur crystals in the protein. And those ferredoxins can transfer those electrons to these enzymes that Roland mentioned in the introduction, the hydrogenases. And you're going to hear more about these enzymes from me as well as from subsequent talks. Okay, so hydrogenases are catalysts that catalyze this interconversion of protons and electrons and molecular hydrogen. Okay, so another way that hydrogen is produced in the living world is through what's called dark or photo fermentation. So these are sort of a couple of different ways combined over here. And this is, again, we need a source of electrons to produce hydrogen. And in this case, the electrons come from glycolysis. Okay, so the breakdown of reduced sugars. That provides electrons. This time they go into reduced molecules, NADH. And from there, they go into hydrogenases, and they're used to reduce hydrogen. Okay, so these are two sort of big categories of hydrogen production in the living world. Another category, interestingly, is coupled to nitrogen fixation. So in the root nodules of plants, where there are the nitrogen-fixing microorganisms, this reaction of N2 and being reduced by 8 electrons for a 6-electron reduction to produce ammonia. The other 2 electrons are going into forming hydrogen. So that's another way that hydrogen is formed very commonly through living processes. Interestingly enough, and you know, if you're forming hydrogen, biology is going to use the hydrogen and use it as an energy source. And sometimes in the same organism and sometimes in different organisms, the hydrogen is used now as a fuel. It's a source of electrons, reducing CO2 to reduced forms of carbon. Sometimes there are other molecules that are the electron acceptors, but carbon is the most common one. So it's a big hydrogen cycle that's occurring in the microbial world all around us all the time. And a lot of hydrogen gets produced and a lot of it gets consumed. And of course, these microbes that consume hydrogen are doing it in a very controlled way to harness that energy. And just as one example, there are microbes that will take the hydrogen produced by nitrogen fixation and react that hydrogen with oxygen to form water. Okay, so those are called null gas bacteria. And those of you who were here last night actually got to witness and hear the null gas reaction, an explosive reaction of hydrogen and oxygen. The bacteria do it without an explosion because they want to harness that energy and do something useful with it. Okay, so I'm now going to talk about these enzymes that catalyze these interconversions of protons and electrons. You can always tell if biology thinks something is really important because it's going to put a lot of energy into doing that thing. And so one of the things that biology has done is developed enzymes that catalyze these interconversions of protons and H2 gas. And these are the hydrogenases. So some general categories of hydrogenases, and I'm going to tell you a little bit more about these. There are nickel-iron hydrogenases, and as the name suggests, these have nickel and iron clusters at their active site. There are iron-iron hydrogenases that have multiple iron clusters at their active sites, and both of those types of hydrogenases catalyze this interconversion. So the reversible reduction of protons to make hydrogen. In some enzymes, this reaction is biased towards hydrogen consumption, and in others, it's biased towards hydrogen production. There's another type of hydrogenase that is known either as the iron hydrogenase or the HMD hydrogenase that has an iron-based catalytic center, and it catalyzes a slightly different reaction, which is shown here. I won't be saying much about this hydrogenase today, but this is an important one, and it was actually discovered in Marburg. So there's a nice paper here by Seigo Shima and Rolf Tauer that describes that. These are some slightly out of date phylogenetic trees of the iron-iron hydrogenase and the nickel-iron hydrogenase. The details here are not that important, but in iron-iron hydrogenase, the red bars are showing where those occur. They're in bacteria and archaea. We now more recently, in eukaryotes, we now know more recently there are some in archaea. And if you can see these colors, you can see that the nickel-iron hydrogenase has a different distribution throughout the forms of life. And this is one of the bits of evidence that these nickel-iron and iron-iron hydrogenases are not evolutionarily related. So these are not enzymes that evolved from a common ancestor and diverged in terms of their metal cluster requirements. It's believed that these enzymes are an example of convergent evolution. So from completely different forms of life, the need for catalyzing these hydrogen interconversions was so important that biology found multiple ways to evolve enzymes to catalyze these reactions. And we can say the same about the iron hydrogenase in that it is not evolutionarily conserved. It is not coming from the same place as the iron-iron or the nickel-iron hydrogenases. So really 3 different solutions to catalyze this relatively simple chemical reaction between H plus and H2. Okay, so now to look just very briefly at the metal centers of these hydrogenases. I'll start with the iron hydrogenase. You can see it's got a single iron. It's got some unusual things bound to it, including carbon monoxide. The nickel-iron hydrogenase has nickel and iron, some cysteines. Again, it's got carbon monoxide and cyanide. The iron-iron hydrogenase has some non-biological ligands that are unusual, including carbon monoxide and cyanide. Okay, so if you know any chemistry, if you remember some chemistry from back in the day when you were studying it, these are all organometallic metal centers. They all have direct bonds between metals and carbon, and that defines them as organometallic. I'm pointing that out because that has been believed to be a pretty unusual feature in biological systems, to have organometallic metal centers. The other thing I'm pointing out, and I've already pointed it out, these all have diatomic ligands, carbon monoxide and or cyanide, bound to the metal. And remember, these are evolutionarily non-conserved, and that means in 3 different instances, nature has found a way to create a metal center that would be a catalyst for this reaction. And in all cases, these metal centers end up having carbon monoxide and cyanide ligands. And that appears to be a very important aspect of the catalytic centers to catalyze these hydrogen interconversion reactions. The nickel iron and iron iron hydrogenases are quite complex. They have a bunch of other iron sulfur clusters that bring electrons into the active site or out from the active site. As I've already alluded to, when biology thinks something is really important, it puts a lot of energy into it. And another aspect of that with hydrogenases is that whether we're talking about nickel iron, which I'm highlighting here, or iron iron hydrogenases, not only does biology need to make the actual hydrogenase and put a metal cluster into it for a catalyst, it has to make a bunch of other proteins just to make the hydrogenase. And this is a process known as maturation of the hydrogenases. So I'm showing you this cartoon here of the nickel iron hydrogenase maturation scheme, basically to illustrate how incredibly complex it is. These different colored shapes are all different proteins. They come together in order to make this protein and to insert the iron and the nickel and to make CO and cyanide and to assemble the active site cluster and to assemble the proteins. It's an incredibly complex process. I'm assuming that I'm out of time since you've stood up 1 more minute. Okay, I just need to gauge what I'm doing here. There's a similar scheme for the iron-iron hydrogenase where many proteins are involved in making the mature iron-iron hydrogenase, whose active site cluster is shown here. And because of the time and because I know Thomas Happe is going to be talking a bit about this in his talk, I'm going to skip over some of these slides. And just, again, as I'm going, remind you or emphasize that the process is incredibly complex just to make these metal clusters in order to make a catalyst that can do this hydrogen interconversion. And everything that you're seeing are things that we have learned over the years about these iron-iron hydrogenases. Interestingly enough, the iron-iron hydrogenase also pulls in some central biochemistry, the glycine cleavage system, in order to make one of the ligands, which is the dithiamethylamine ligand. And that's something that we've discovered quite recently. So to sum up what I've talked about, too slowly, obviously, is that hydrogen is really a central player in biology, particularly in microbial biology. It was a key player in the emergence of hydrogen catalysts in the early Earth, the emergence of life in the ancient forms like archaea, and modern forms that are playing an important role in the hydrogen economy around us in the living world. And with that, I'm just going to end and thank the people in my lab and collaborators who've worked with me on some of the work that I've talked about. Thank you.
Thank you, Joan, for this wonderful presentation. We still have time for 1 or 2 questions. Please.
We heard this morning about hydrogen exoplanets. So would be the question, so Lisa Kaltenegger is always asking that, is there life outside our planet?
Yeah. I wish I knew the answer to that. I mean, I think, no, it's a great question to sort of tie together this morning and this afternoon, which is that hydrogen can be a major force in driving life, the chemistry of life, the biochemistry of life. And so hydrogen is obviously very abundant in the universe. It's a fantastic source of energy. And I think, I mean, more than likely than not, there are other forms of life out there, and hydrogen is probably playing a role.
Please.
Many thanks for this nice presentation. I'm really fascinated by these microorganisms or archaea, methanogens, and I was wondering, did you have any ideas to use it also for a kind of commercial way to produce clean hydrogen, or methane as a kind of energy source? You have already plans? I don't know.
Yeah, no, I really appreciate that question. I mean, one of the reasons we as biochemists are interested in hydrogenases and or the microorganisms that make hydrogenases is that there is a potential to be able to use these as a way to produce hydrogen in a green way, in a way that's not going to require massive amounts of electricity, for example. And so there is a lot of interest in that. There are people that are engineering these microbes in order to make them better at capturing sunlight and converting that into producing hydrogen or other types of reduced molecules. So it's a great question, and yes, yeah.
Yeah, thanks, Joan, for this wonderful presentation. And I guess this question was the nice start for the next two speakers, because probably they address a little bit of your question. The next speaker is Mirko Basen from the University of Rostock, and I'm especially grateful to Mirko because he jumped in on very, very short notice for his former mentor, Volker Müller, who for health reasons couldn't make it, unfortunately. We wish him from this stage here all the best for a quick recovery. So Mirko studied biology at the University of Konstanz and then worked mainly in Bremen with Professor Widdel and got his PhD there. Went to the University of Georgia in working with Mike Adams. Then went to the Goethe University in Frankfurt working with Volker Müller. I mentioned this already. And since 2019, he was a junior professor at the University of Rostock, and 2023, he got a full professorship. His interest is in acetogenic bacteria and energy converting hydrogenases. And I think he will also tell us a bit how we can use hydrogen for good purposes. Mirko, thanks.
Yeah, thank you so much, dear members of the Leopoldina, dear audience. It's a great honor for me to be here instead of my former mentor, Volker Müller, who sends his best wishes and also provided me some slides which hopefully convince me in the next, will help me to convince you in the next 20-25 minutes that microbes are very, very important in hydrogen cycling in anoxic environments, and they also might provide you some solutions for hydrogen bioeconomy. So I will start with a different gas. So in Rostock, in northern Germany, we don't only have the Baltic Sea, which is beautiful, but also lakes. And this is actually a lake near to a cafe. And I was sitting at the cafe table, and then I grabbed a stick, and let's see how that works to start the video. And this is called the Volta experiment. So if you have a lot of biomass, you saw all these reeds, and you poke in these organic-rich sediments, gas bubbles out, and that gas is not hydrogen. It's likely about 70% methane, and the rest is, most of the rest is carbon dioxide. And Volta actually caught that and showed that this is inflammable gas and discovered methane. So how is that methane produced? We heard a little bit already in the last talk. It's basically produced in anoxic environments when biomass, here, for example, plant polysaccharides, are broken down by different types of microorganisms. And in the end, you will yield methane and CO2. And hydrogen is a very important intermediate because it's actually the substrate. Hydrogen and carbon dioxide are the substrate for this group of microorganisms called methanogens. And the biochemistry of this has been in detail studied and elucidated by Professor Tower, who is in the audience. But you see, hydrogen is an important intermediate here. And if you look, and this looks very complicated now, in anoxic environments, two gases, but CO2 is not the topic of today's talk. Hydrogen and CO2, they are very important intermediates, and they are produced by fermentative microorganisms next to different acids, and then there are different cohorts of microorganisms that actually, or groups of microorganisms that consume the hydrogen. If you have sulfate, like in marine sediments, sulfate reducers, and then these two groups, acetogens and methanogens, and that actually convert hydrogen and carbon dioxide to methane and acetate. And these groups of microorganisms actually keep the hydrogen partial pressure in these anoxic environments pretty low. It's between about 1 to 10 Pascal, so it's pretty low in the micromolar range. And this enables actually fermentation to continue. And despite that, the turnover rates are, of course, pretty high, and these organisms are pretty important for the environment. Also in our gut, I was actually surprised to read that if you take the off gas of our gut, also in our gut, of course, there's microorganisms that ferment the polysaccharides or saccharides that we eat and produce hydrogen and carbon dioxide, and the average hydrogen concentration in the off gas is about 3%. And it's highly variable among individuals and might reach up to 86% at some times, depending on what you eat. And of course, the microorganisms are influenced by the hydrogen and influence the hydrogen concentration, and also they need to recognize the hydrogen. That's also often forgotten, so hydrogen sensing happens there, and actually your former Vice President Bärbel Friedrich was the first to describe a hydrogen sensing hydrogenase. And hydrogen in our gut also influences our health. It is negatively correlated with certain diseases like inflammatory bowel disease, infections, and even colorectal cancer. So hydrogen is obviously good if you have hydrogen in our gut. But how is hydrogen actually formed? It's formed by fermentative microorganisms, and fermentation actually was described by Louis Pasteur as life without oxygen, but maybe the more precise current definition is fermentation is a biological redox reaction for energy conservation without an external electron acceptor. So in these anoxic environments, when you also have no other electron acceptor like sulfate, but no oxygen, these organisms have to come up with a solution, and what they do is they use sugars and partially oxidize them, this is glycolysis, and that yields ATP. But then they have a problem, they have to get rid of their electrons. And so nature invented soluble electron carriers like NADH, and NAD is then reduced to NADH, and then the NADH, the electrons are recycled by internally, these organisms create their own electron receptor like pyruvate and produce lactate. That's actually what happens in yogurt, for example. And the problem is here that in this recycling of electrons, you get no ATP. Compared to our metabolism and the metabolism of oxic or aerobic bacteria, that's very different because their glucose is by the further steps that you all know from school knowledge. Pyruvate is further oxidized to acetyl-CoA. That yields additional electrons, and then acetyl-CoA is in the citric acid cycle also oxidized yielding even more electrons transferred mostly to NADH. And this NADH goes to the respiratory chain, where the electrons are used to pump protons out of the cell, and then the protons can be used to fuel ATP formation. And this process called electron transport phosphorylation yields most of the ATP in aerobic bacteria and also in our own metabolism. So fermentative microorganisms have two problems. Actually, they conserve much less energy because they don't have oxygen or any other electron acceptor, and they must get rid of electrons. And here's another solution to get rid of electrons. You can also use pyruvate, the end product of glycolysis, decarboxylate that oxidatively, releasing another pair of electrons, and then that yields acetyl-CoA, and acetyl-CoA can be reduced to ethanol. However, the acetyl-CoA, this yields no more ATP, but also the acetyl-CoA, and that's another option that fermentative organisms have. They convert acetyl-CoA to acetate, and that actually yields 2 additional ATP. And that's what the fermentative microorganisms want. They want additional ATP. But then they have the problem that here's NADH, and here's 2 electrons from pyruvate oxidation, and they have to go somewhere, and the best would be actually to produce hydrogen, because then they solve the energy problem. They get more they get more ATP, and they solve the electron problem because hydrogen can be blown out. The problem here is that NADH it has a redox potential at pH 7 of the pair NADH to NAD of minus 300 millivolts, and that is actually not sufficient to reduce protons to hydrogen. 2 other electron carriers, formic acid or formate, which I will tell about later, and ferredoxin, a small redox active protein containing iron sulfur groups, actually they are reduced enough to reduce protons to hydrogen. And that is also carried out by biology. So here's an example of Escherichia coli, a common gut bacterium, which does a so-called mixed acid fermentation. It gets rid of its electrons by producing certain acids, like succinate or lactate. And then also it converts pyruvate to acetyl-CoA and formic acid or formate. And then this formate can be used to reduce hydrogen, protons to hydrogen, and releasing CO2. And that enzyme is called a formate hydrogen lyase. It's a membrane-bound enzyme consisting of a formate dehydrogenase subunit, electron transferring subunit, small here in orange, and a hydrogenase subunit. It's a membrane-bound enzyme that actually has been studied here in Halle by Gary Soers and Cornelia Pinske. And the second electron acceptor that's reduced enough is ferredoxin, and that is actually what butyric acid producing Clostridia do. They oxidize pyruvate to acetyl-CoA, and the electrons are transferred to this ferredoxin, and then the ferredoxin is used to reduce protons to hydrogen concomitantly to the butyrate production. So now we have a completely different organism, which is much more ancient, which is called Thermotoga maritima. And Thermotoga maritima grows optimally at 80 degrees Celsius, converting sugars to acetate, CO2, and hydrogen. So actually this organism exclusively produces hydrogen and no other, like no ethanol or so, no lactate under certain conditions. And it reaches a so-called tower yield of 4 hydrogen per glucose, which is the maximum amount of hydrogen that you can reach in dark anaerobic fermentation, because fermenters do not oxidize the acetate anymore. And how does the organism do it? That's a problem because in glycolysis NADH is produced, and pyruvate oxidation yields ferredoxin. So the ferredoxin is reduced enough to produce hydrogen, but the NADH is not. And again, this is a discovery of Rolf Tauer. The solution is called electron bifurcation. And now I tell it the opposite way. So here you have hydrogen as electron donor, and hydrogen as electron donor is not reduced enough to produce ferredoxin or reduce ferredoxin, you need energy for that. However, if you couple that oxidation of hydrogen to the reduction of NADH, NADH is a very good electron acceptor if you oxidize hydrogen, that releases energy, and if you couple these two reactions, you basically, one electron can go to NADH, and the energy is transferred, and this enables one electron going uphill to ferredoxin. This is likely how ancient anaerobes produced ferredoxin. They need ferredoxin for many processes, and it's not a mechanism of energy conservation, but it spares them some energy. So basically, these enzyme, in this case, it's an electron bifurcating hydrogenase. If Professor Müller would have been here, he would have shown you the crystal structure of this enzyme from Acetobacterium woodii. This enzyme either enables hydrogen oxidation and coupled to ferredoxin and NADH reduction, or NAD reduction, or it enables also that all electrons from NADH and ferredoxin go to hydrogen, which has originally described by Mike Adams. And this can be exploited because there's actually one group of microorganisms, anaerobic, extremothermophilic, growing optimally at 78 degrees Celsius. They're called, this organism is called Aneurocellum beskyei. Formerly it was Caldis cellulosiraptor beskyei, the one that at hot temperatures disrupts cellulose. The organism can grow on unpretreated biomass, biocrops that are studied in the United States, like switchgrass or poplar. It secretes enzymes to degrade this biomass and takes up the sugars and converts the sugars to acetate, CO2, and hydrogen. And it also yields a lot of hydrogen. So potentially these organisms can be modified to as highly efficient biomass converting organisms. However, there's still the problem that most of the electrons are still stored in the acetate because fermenters cannot, or fermentative microorganisms cannot oxidize the acetate anymore. So is there a way to increase that? Yes, there might be ways, and one is that, and that has been also experimentally shown, and potentially the next speaker is going to talk about that. You can couple dark fermentative hydrogen production with the removal of acetate by acetate utilizing phototrophs, which is called photofermentation. Another option, which has not been studied that well, is that you could also use acetogens, the group that I'm going to talk about next, and to try to get them not to produce acetate, but oxidize acetate to CO2 and hydrogen, and technically remove the hydrogen, which is called hydrogen milking. So now we are at the acetogens, and I think I have a couple of minutes left, and this group is of very high importance in the environment because it uses the hydrogen intermediate and CO2 to reduce that to acetate, providing the acetoclastic methanogens, a group of methanogens that basically split acetate to methane and CO2. On the other hand, they are also opponents of the methanogens because they compete for hydrogen and CO2 with the methanogens that actually feed on the same substrates. So they're defined as a group that thrive, of microorganisms that thrive on hydrogen and carbon dioxide conversion to acetate, but many of them are metabolically versatile and can also ferment sugars and alcohols to acetate. And I'm not going to go into detail here. This is a picture of the best studied acetogen, Acetobacterium woodii, where Professor Müller elucidated the whole bioenergetics. On the right side in green, that's the famous Wood-Ljungdahl pathway, which takes 2 molecules of CO2 to reduce them to 1 molecule of acetyl-CoA, and that's actually what they do as their respiration. So they stoichiometrically convert CO2 to acetyl-CoA, so 2 times C1 to 1 times C2. And they're the only group of microorganisms that does that stoichiometrically. And what I want to point out here is that a very important intermediate is formic acid or formate, and this reaction here will become important in a second. But of course, acetyl-CoA is also a building block of cells, but also potentially a building block for organisms to produce other compounds out of this C2 compound. So these organisms are of biotechnological relevance. Interestingly, Acetobacterium woodii has two hydrogenases, and one is this electron bifurcating hydrogenase that oxidizes hydrogen, yielding electrons in form of the ferredoxin and NADH as electron carriers, which are then reoxidized in the Wood-Ljungdahl pathway to, in the end, form acetate. But also the first enzyme here that reduces CO2 to formic acid, actually here should be hydrogen. It can also use ferredoxin, but the main electron donor for this enzyme is hydrogen, and it's the first soluble hydrogen-dependent carbon dioxide reductase. It consists of a hydrogenase subunit and then connected to a formate dehydrogenase via two small subunits that transfer the electrons. And this enzyme is of interest for different reasons. So first of all, in the metabolism, environmentally, if you grow Acetobacterium woodii on a sugar, it actually will produce NADH and ferredoxin like thermotoga. And then this electron bifurcating hydrogenase operates in the confocating direction, unifying, channeling both electrons to hydrogen. And then the hydrogen actually is used by the same cell by this HDCR enzyme to reduce CO2 to formic acid. It's not a hydrogen cycling between different organisms, but in one cell. And then for structural reasons, this enzyme actually forms long filaments. The longer the filament, the better the activity. And here you can see in the middle, these are the small subunits. And here's the formate dehydrogenase. On the right side, there's the hydrogenase subunits. The longer the filaments, the more active the enzyme is in that case, producing formate from hydrogen and carbon dioxide. And then Professor Müller, together with collaboration partners, studied the structure and found that actually the electron transferring small subunits basically connect the hydrogenases here shown in blue, two hydrogenases subunit per small complex, and one formate dehydrogenases connects these subunits, forming a long nanowire structure. And basically the idea now is that hydrogen at different points of this nanowire can feed in electrons, which are then transferred to reduce CO2 to formate. But the enzyme also can operate in the opposite direction, oxidizing formate to CO2 and reducing protons to hydrogen, like an electron autobahn. And potentially this enzyme can also store electrons. How to proceed? And then I looked into the cell, and then in a second you will see in yellow that the enzyme not only forms long filament, but also bundles of filaments attached to the upper cell pole in yellow here. The blue are ribosomes, like curtains dropping from one pole of the cell, so the organism actually has a lot of this enzyme, and we are not sure yet why actually the enzyme is attached to the membrane, because it's a soluble enzyme. And now here to the right, the pure enzyme actually, and that's the third reason that it's interested, has a very, very high activity. Here, the HDCR of Thermoanorobacter kivoi, which is a thermophilic acetogen, has turnover frequencies in the hydrogen production direction as well as in the formate production region of 10 millions. It's by far better than any chemical catalyst that's known. And that brought up the idea to use not only the enzyme, but the whole cell of Thermoanorobacter kivoi, but also of Acetobacterium woodii, which also had reasonable activities, and block the formation of acetate only to use cell suspensions to reduce CO2 to formate with hydrogen. And Professor Müller has shown this in kind of like a biobattery. You can, in that case, this was done by genetically engineering the organism that it couldn't, under certain conditions, couldn't produce any acetate. And then he introduced day-night cycles. So during the day period, he fed in hydrogen and carbon dioxide, and in the night period, the culture was flushed with N2. And here on the top left, you can see the product concentration of formate, which in the day cycle reaches concentrations of 25 millimolar. In the night cycle, it goes down again, and the enzyme could do that like for several cycles. And now you can imagine that this can be employed maybe even on a household scale with a little bioreactor in the house and solar panels on the roof, which are then used for electrolysis, providing hydrogen, which is converted biologically to formic acid, the liquid organic hydrogen carrier, and then the hydrogen in the night period is released and used for energy process. And on a bigger picture, these organisms then can be used to convert electrons or hydrogen from biomass, synthesis gas, industrial off-gas, via organisms that contain this HDCR enzyme to formic acid, and the formic acid can then either be transported as liquid organic hydrogen carrier, chemically converted, or microbially converted to other products. And that's actually what my lab is interested in. And in a BMBF-funded project called Thermosynthon, we were using Thermoanorobacter kiwii, the thermophilic acetogen, to utilize synthesis gas mixtures to produce compounds. And actually, Thermoanorobacter kiwii is by far the fastest acetogen. At 66 degrees, it grows as a doubling time of 1.2 hours on hydrogen and carbon dioxide. Also fast on synthesis gas. We have a genetic system. For example, if we delete certain nickel-iron hydrogenases, it makes more acetate. But in the project, actually, we wanted to produce something else. So we introduced the pathway for ethanol formation with a very special ancient enzyme called AOR, aldehyde ferredoxin oxidoreductase, and we succeeded in proof of concept that the organism actually produced ethanol now. Of course, the concentrations are not that high yet, but we are working on improving that by genetic engineering and in the bioreactor. And this process actually, we are in the footsteps of a company called LanzaTech that actually uses these acetogenic microorganisms bioeconomically. They have 5 operating plants. Again, Professor Tauer, I think, is in the board of these company, and they produce ethanol, which is then further converted and derived of CO2 from renewable sources. So, and I hope that I convinced you now. I'm not going to go through all of my summary that hydrogen is an important intermediate in anaerobic degradation of biomass. The concentrations are quite low, but there's several types of organisms that utilize this hydrogen, and potentially, for example, with the last part of my talk, there might be biological solutions for a hydrogen-based bioeconomy, and these catalysts, these enzymes, they need moderate conditions for hydrogen production, storage, and valorization. And with that, I would like to thank my laboratory and also my collaboration partners. And of course, Professor Müller, who provided a lot of the slides.
Yeah, thank you very much, Mirko, for this wonderful presentation. Questions for Mirko? Yeah, please.
So, if you want to go into the production, so what about the longevity of the system? So how long can you really produce dihydrogen and the other products, or ethanol and whatever?
That is currently studied. Like in the slide that I showed you, you saw that it extended for a couple of 100 hours with the bio-battery system. And currently, a South German company, Festo, they have a collaboration with Professor Müller using Thermoanorobacter kiwii to produce hydrogen, formate from hydrogen. And they capture, I think they capture the formate in their bioreactor. So it's like the economical side or more industrial side that's currently under investigation.
Mm-hmm. Further questions?
But it's better to use whole cells than enzymes.
Yeah, this would have been my question. What about enzymes and putting it into a reactor and keeping the enzyme stable, because then you would avoid the cell reproduction?
Yeah, my question would, or my answer would always be, it's better to keep the cells, because the cells can recycle and produce more enzymes. I don't know if, I think there was one collaboration, but that Professor Müller could have told you where you basically fix this HDCR enzyme on an electrode. But that's of course a very small scale. So if you're talking like enzyme applications for these like large scale processes like formate production, that's likely a question of really of upscale. And with bioreactors you can do upscale, like with whole cell catalysis.
So most likely the audience asks themselves how much you can produce in a normal reactor.
So acetate, which is also of value, can be produced up to concentration of 500, 600 millimolar, which is maybe not exactly industrial scale, but for microbiologists there's already a lot. And I think formate concentrations reach up to 600 millimolar. And if you scavenge that by technical solutions, you can drive, like we can continue with that reaction. And that's actually the idea.
Okay. Thank you very much. I don't see further questions. Then we come to the final presentation of this biology section, and I call upon Professor Thomas Happe. He's from the University of Bochum. Actually, Thomas studied biology in Bochum with Professor Trebst, and then went as a postdoc into biotechnology at the University of Bonn, spent some time at the University of Berkeley in California, and learned the use of Chlamydomonas. We heard about this algae already, to switch from the production of oxygen to produce hydrogen. I think you will touch upon this. And what Thomas most likely will also touch is that we can take the cofactors out of these enzymes and use them for catalysis in the idea of producing hydrogen. Thomas, thank you for coming.
Yeah, thank you, Roland, for this kind introduction, and I hope I can convince you at the end that also enzymes are a very fascinating possibility to produce hydrogen, and not only cells. Okay, I do my best. And to begin with, I would like to explain you what I understand as a biologist for green hydrogen, okay? Now we are talking about green hydrogen, and you have heard that you can use electricity on an electrode, on a platinum electrode or whatever, to split water in hydrogen and oxygen. In nature, you find organisms that can do the same. So for instance, here you have does not work, the laser pointer. Anyway, you see this tiny small organism, Chlamydomonas reinhardii. These are eukaryotic green algae. And you see also the green bottle. Okay. And you see only you see also the green bottle, and might be you see also the bubbles here on the top, and the bubbles are pure hydrogen produced by the organism, because they can split the water to hydrogen and oxygen via metabolic processes. And Tassius Melis, a good colleague and friend of mine, has discovered this process. You might know that green algae and also plants can do photosynthesis, so they use the reductive power from the light. I have problems with the laser pointer. Anyway, so they can use light, split water, take the electron, and produce biomass like starch or sugar. And under special conditions like stress, they change the metabolism and do not produce any biomass anymore, but give the electrons to protons and produce hydrogen. And you can use, in principle, the hydrogen for sustainable hydrogen production. Unfortunately, the process is not stable enough. And we have analyzed this a lot. You see on the left side a big photoreactor producing hydrogen, but our dreams that green algae can produce fuel for the future did not come true at the end. So we switched to enzymes, to proteins. Okay. Perhaps it's empty, I don't know. Okay, so now I want to switch to enzymes, proteins, and they can catalyze this simple chemical reaction, proton, electrons to hydrogen that we heard a lot. And I will focus during my talk especially on iron hydrogenase. So this is a special class of hydrogenase. So you see here the enzyme consists of a protein envelope. So these are amino acid sequences, so around 400 to 600 amino acids. This is a protein envelope, but this cannot catalyze the hydrogen production. But you have here this organometallic cofactor that Joan has spoken about. And this is very important for catalysis, okay? So therefore I have to explain it a little more in detail. Here on the left side you have a iron-sulfur cluster of 4 iron and 4 sulfur, and this cluster is needed to transport the electrons. And on the right side you have a special unique cluster of 2 iron, 2 sulfur, and CO and CN ligand. And here at the distal iron site, this is a catalytic site where the enzymes can produce hydrogen. And this they are doing in a very special way, even for chemists, because they are producing a hydride ion. And in the beginning, when I talked to chemists, they told me it's impossible. A hydride ion is not stable enough. So, but we find out that at this position, first a proton bind, then it's twice reduced to a hydride ion, and then a second proton comes and the hydrogen molecule is released. And the turnover frequencies that you heard are extremely high. So I have written here that one molecule of the hydrogenase can produce in a second 10,000 molecules of hydrogen. So also for application, very, very interesting. We have only one problem because the enzymes are very oxygen sensitive, but I have a solution for that later. So a short explanation why we have such a high turnover frequency, because we have a so-called proton-coupled electron transport. So we need protons and electrons, so we have defined channels where the protons come from the bulk solvent here, are transported through several amino acids to the active cluster, and also from the left side, the electrons are transported via an electronic relay. And because we have a concerted action, this is quicker than the action than any action alone, than the electron or the proton transfer, you have a very, very high production rate. Okay, coming back to the cofactor. Unfortunately, Joan has not so much time to explain this really, really fascinating maturation process. I have only one slide, sorry for this, Joan, but it's very important to understand that in nature you have 3 additional proteins called Hg, E, and F that are producing this H cluster. And this was analyzed nicely by Joan. Joan is really the expert in this field. Okay. But you have to know that this special cofactor is formed only in specialized organisms. And if we come back later to application, we need a good organism where we can overexpress our enzymes. And the typical organism is E. coli. I think every one of you know, if you produce proteins, you use E. coli, okay? But E. coli cannot express the active cluster. Okay, what can we do? So we can look again at the structure, and over 25 years ago, John Peters has solved the structure of the enzyme and also of the H cluster. And during that time, the chemist recognized immediately that the H cluster looks not so difficult for a chemist. And indeed, Thomas Rauchfuss, 1 year later, synthesized already this cofactor in a gram basis. So he told me it's very easy for him to synthesize this cofactor. Unfortunately, the cofactor alone is dead. So again, in the beginning we thought this is a solution of our energy problem, but the cofactor alone cannot produce hydrogen. Okay, what can we do? So I think everyone sitting here, every scientist have once in his life a moment where he make his or her experiment. And this experiment is I call it Alexander Fleming experiment, because it's easy and so it's so unexpected. So we have the idea in Bochum, so okay, we have the protein that we can easily express in E. coli, in gram. We can easily express the cofactor or synthesize the cofactor. What if we bring them together and just mixing the two components and see if we get an active enzyme? So the idea is so crazy that our colleagues, that even my PhD student said, we will not do that, okay? So, but at the end, I can convince my students to do the experiments, and we are right. So here's a typical result. So the complex alone has no activity, the enzyme alone has no activity, and just by mixing for some minutes, we get a very high activity that is even higher than in the natural organism. And this was a big breakthrough, because you can imagine that now you can, in principle, mix any chemical component with any protein, and look what happened. Yeah, so far, under that time, no one has really think about that. And my colleagues called this interpretation here a dance between a metallocluster and a protein. So we have the cluster that is dead, and by incorporation into the protein, the symmetry, the nice symmetry of this cofactor is destructed, and we get an open coordination site where the proton can bind, and we have activity. So at the end, very simple, you have to try this. So the next step was to produce the enzyme on a huge level, and we succeeded it. So when we start during my PhD, I could isolate 1 microgram from an organism per liter. Nowadays, we succeeded in getting 1 gram per liter. And this is huge. You can imagine that you have now a lot of protein that you can use also for application. What is also very interesting, the enzyme is extremely stable, more stable more or less than any other protein. So some tests. So we tested with pressure, yeah, at 2000 bar, the enzyme still produces hydrogen. We look at temperature. We have a temperature optimum at around 60 to 70 degrees, so it's also quite nice. The next thing was looking for organic solvents. So normally protein are degraded in organic solvents. For instance, for application, if you want to use them in acetone or acetonitrile, they are stable, no problem at all. In alcohol, we have some problems, but in most of the solvents, they are quite stable. And there was a question from the audience. The hydrogenases are also stable during production. Here is a study on, if you put the hydrogenase on an electrode, we recognize after 7 days that we still have the same activity then as in the beginning. Okay, so we have a very stable enzyme against everything. We have only one little problem, that is oxygen. And we can analyze this very easily. So with several methods, here I bring you with me the direct method. So we look on the structure of the cluster, and we recognized here this red balls are the electron density of the iron atoms. And if you have balls, then you still have an iron atom, and if not, you have no iron atom. So you start the experiment without oxygen, and you see in the distal cluster, this is in the electron relay cluster, you have fully occupied cluster. The same is with the H cluster. After incubation of 23 hours, in the control, you still have the full cluster, but you see in the H cluster, you lose the density, meaning the cluster is degraded. So this is a clear hint. If you put the enzyme into oxygen, the enzyme is degraded because the cluster is not there anymore. Okay? What is the solution? You can look in nature. Okay? You can look in nature if you find an oxygen-tolerant hydrogenase. Okay? That was done already 10 years ago by an Italian group of Simone Mora, and he recognized, in comparison to the standard hydrogenase, so here's a standard hydrogenase. This is an infrared spectrum. These are vibrations of the CO and CN bands. You see under normal condition, you have nice bands, CO and CN bands. Under oxygen, nothing. showing the enzyme is dead. If you do this with another enzyme from a Clostridium species, you recognize that under oxygen, you see still the bands, meaning the enzyme is still there. But you also recognize that there is a shift of this band to this band, and this means that under this condition, the enzyme is active, but under this condition is not active. So it's inactive in the presence of oxygen, but it can reactivate it, and this is very important, of course. So you can somehow store it under oxygen. And the reason for that we find out because you have a protection of an amino acid. This is a cysteine ring here, a cysteine. This protects this distal iron. So again, you have the control of the model enzyme, and you see that there is no cap. We called it safety cap mechanism. So there is no cap that this cysteine can protect the active cluster. In the case of the CB5H, the oxygen-tolerant enzyme, you recognize that you have a protection of this cluster under oxygen. Still, it's not active under oxygen, but it's protected. And the reason is because the enzyme is extremely flexible. Yeah, you have to imagine that he can change the structure very easily because of several amino acids. And this is also quite interesting. Okay. We solve also the cryo-M structure. We know everything now very clearly that also for the specialists, a zinc atom is included into forming the dimeric structure, but this is only for specialists. And this is the mechanism here that we can shelter our enzyme, our new enzyme against oxygen. However, at the end, we discover that the enzyme although it's stable under oxygen, if you repeat the mechanism, so without oxygen, and so on, they lose activity. And this is also not quite good, okay? We need another solution. Again, we go back into nature. And in nature, we find now a really, truly oxygen-stable hydrogenase. And this I have never presented on any other conference, and this is really new, and this is really sensational, because for the first time, we have a very stable, truly stable hydrogenase. So I will convince you. So the first experiment that we have done, we leave it on the bench. So in this experiment for 7 days. So we isolate the protein under aerobic conditions. We leave it on a bench for 7 days, measure the activity, and it's still active. So it looks not so sensational for the physics, but for the biologists, I think it's really quite unusual that we have really a truly high, total stable hydrogenase. And it comes even better. So if we test the activity under oxygen, yeah, what happens? So we have anaerobic conditions. This is a normal test assay that we use. We have our control enzyme activity. We have our new enzyme. We have the same activity. And then if you test the reaction under oxygen, you have to imagine that we measure the hydrogen production under oxygen with an enzyme that are normally oxygen sensitive. We can show that we have hydrogen production. And this is for the first time that is really 100% stable hydrogenase. And I think this will change in the near future everything. Okay, but we still have problems. We are also working with other systems to test if that are perhaps more easy to produce hydrogen. And the biggest problem is, of course, we heard it yesterday from Freddie, where come the electrons from? Yeah, if we want to produce hydrogen, we need electrons. So you can use in such a system, for instance, chemical substances like photosynthesizer. You can use light here. You can put even the enzymes on electrodes and test and use electricity. But you can also use natural physiological donors. You have heard from Joan already that ferredoxin is a natural electron donor for the hydrogenase. And one idea that we have is, let's try our system to include the inactive cluster into a ferredoxin. Again, a very crazy idea for a biologist. But it turns out it works. Okay, so we integrate the cluster into a very small protein, only 100 amino acids long, and we see that we have activity. And this is quite nice because we can now use the natural system and fix the ferredoxin to the photosynthetic reaction. So in a normal way, you have a very complicated system. You have an electron donor, you have a hydrogenase, a big protein, and so on. With a new system, in principle, we can directly interact the ferredoxin with a photosystem. Also a very good, nice application for the future. And we want to go the next step again. So we also want to integrate the cluster not only in peptides or proteins, but also in DNA. You might know that DNA is a very stable component in nature, and you might know that there are also some DNA enzymes available, and we want to try if it's possible some in the near future if we can integrate our cluster and have activity. I cannot promise that it will work out, but what we see so far is that we can integrate the cluster in a small DNA structure. We call it aptamer, and this seems to work as well. So we have a very, very good and very interesting cluster that can be used for several purposes. And because all of you want to know how efficient is the system, I don't like it, it has to say, as a biologist. I'm a basic researcher, but nevertheless, I bring some calculation with me to convince you that this enzyme system might be also interesting, even for the hard chemist here in the audience. So there was a famous publication, 1999, in Nature from Dick Chemick. Dick Chemick is a very famous chemist in Imperial College in London. And he made an extrapolation saying that if we would have 1 mole of hydrogenase, it could be possible that we can fill an airship in 10 minutes. Okay, this is huge. This reminds me of a big hydrogen plant. And I don't want to talk about big hydrogen plant, but let's talk about a car. You know, perhaps some of you already have driven a hydrogen car, and to drive 100 kilometers, you need 1 kilogram of hydrogen. The question is it impossible to produce this? And the answer is yes. For this, you need, in principle, 3 grams of the hydrogenase. This means that we need only 10 liters of E. coli, and in 10 minutes, you can produce enough hydrogen to drive a car for 100 kilometers. Okay? Of course, we need the electrons. This is the bottleneck, still the bottleneck that you have nicely from Freddie yesterday. But in principle, you can use this hydrogenase now also to create an electrode or something to split water, whatever you want. With this, I'm at the end. I like to thank my team, and my team is also very active during the Corona crisis, and I'm very happy to answer all the questions that arise now. Thank you very much.
Yeah, thank you also for guiding us into potential applications. I think you feel that there is still a lot to be done to make it usable, but we are on the right way. Questions? So, okay, please.
Thank you very much. I basically don't understand anything. I'm a psychiatrist, but I came here because I'm really interested in this. I don't like electric cars for whatever reason, for many reasons anyway. So I'm really hoping that one day I can drive a hydrogen-powered car. So, and this is now not a scientific question. This is more a question. So you said it could be done, but
you need a fuel station, right?
The fuel,
you need a fuel station in your nearby your house.
Is it a lack of political will, societal will, or that could we do more? Could we push that more? I mean, we seem to be pushing electric vehicles.
The problem is the price, right? Mm-hmm. To be honest, it's always. Yeah. I mean, if you want to pay 10 times, 20 times more than for regular hydrogen, we can make it. But yeah.
Yeah, I hope maybe at the end of the day, more solutions.
Please, over here.
Yeah, that's actually a very interesting question, and I'd like to comment on that, because the first question is, do you in the future see that you would drive your own car? So that's the first question, because thermodynamically it's maybe not the best solution to have a, let's say, 80-kilogram human in a 2-ton car. So the next thing is, what would it be fueled by if you drive a car that is not your own car? Is the car the best vehicle? Is it a bus? Is it a whatever, you know? An autonomous little thing or so. And when it is fueled by something, what is the cost in total for the full cycle to actually make your fuel? And I think at the moment, we need the hydrogen for other things.
Further questions?
Yeah, very interesting talk. So I just wonder if you can couple this enzyme hydrogen generation with the electrochemistry together, and will be able to show at the demonstrator at the electrolyzer with a very high efficiency at this stage?
Yeah, so this is a big field at the moment, so several people working on that, or not several, a lot of people working on that. The problem was the stability so far of the hydrogenase and the oxygen sensitivity. And I think we have now solved the problem, and yeah, let's see what happens in the next years on this field. Thank you.
You need a neutral edge condition to be compatible with the biology system, the biocompatibility.
Of course, yeah.
But in the long run, how stable is your enzyme over weeks, months?
Yeah, I have shown you some experiments. So the enzyme itself, it's stable, I would say, over years.
Really?
But the activity, this is a question, right?
Yeah.
So, I mean,
that's what I
but
mean.
there was a study showing that for 7 days it's possible to produce hydrogen on the same level. So I don't know what happened over the next years.
For production purposes, this is of course not enough to have it just for a week, you know.
Yeah, of course.
You need it very long time stable.
But we have, there was a question if we have enough money.
Questions? So that's further questions? We still have time, but if not, then we can hand over to Peter and the next speaker a little bit before time. Thank you very much for the biology talks.
Yeah, we're changing pace a little bit. My name is Peter Schreiner. I'm a chemist, and we're changing towards chemistry in physics again. Currently, I also have the role of speaker of the chemistry section of Leopoldina, and I have the pleasure of introducing our next speaker, Beatriz Roldán Cuenya, who is currently director of the Interface Science Department at the Fritz Haber Institute in Berlin. She began her career in Spain, got a master's degree in physics, and later a PhD in physics in Germany in 2001, did postdoctoral research in the US at the University of California, Santa Barbara, and after that immediately joined another US institution, the University of Central Florida, as an assistant professor and rose through the ranks to become a full professor in 2012. Immediately thereafter, she became a chair professor for solid-state physics at the Ruhr University in Bochum, before she moved in 2017 to the Fritz Haber Institute in Berlin. She has garnered quite a few accolades. I'm not going to list them all. I'm just going to point out two of them. That is the Margarita Salas Award in 2024 and the Faraday Medal of the Electrochemistry Division of the Royal Society of Chemistry in 2022. And last but not least, she's become a freshly minted member of Leopoldina last year, as we learned yesterday when you received your plaque. So welcome. Stage is all yours. The title of your lecture tells us there will be no future without hydrogen, fundamental aspects of synthesis and application of green hydrogen. Please.
Thank you very much for the introduction. It's a pleasure for me to be here while we are getting the presentation ready. So I think it's going to be a drastic change with respect to the other talks of this afternoon. So I'm not a biological person, but I'm going to be talking about catalysis also, and why does this matter? Yeah. So the first things to consider is that we know right now that fossil energy resources will run out at the end of the century. So we need an alternative. If we are thinking about where do we get the energy from, so we know that we can produce a lot of electricity from the sun, so 4,600 terawatt per year that will fall on land, and mankind only needs 19. So actually we have enough energy. if we are thinking about sustainable options, we need to go away from nuclear and carbon capture and storage because we need to close this energy cycle. In energy, we are talking a lot about whether we can go, we can have all our energy supply just with renewables. And if you look here at the energy demand, just for January last year, you can see what we can produce with solar in Germany, wind, biomass, etc. And then you can see that unfortunately there are something that we cannot cover. And this means that we have volatility of the primary electricity, we have an unbalanced distribution of the sources and demand, and the power supply will need that we have solar fuels. This is something that I will be illustrate today for combustion on demand. We talk about hydrogen, so this is my hydrogen map here. The color here in blue represents how expensive is making hydrogen in certain regions of the planet, and in red is where it will be the cheapest. And you can see that in Europe, the country where I come from, in Spain, that's where we can produce at a relatively okay price the hydrogen. Of course, North Africa, Australia, and other parts of the world. But you can see that there are many places where you can make the green hydrogen. So we have global availability. I changed my talk after the talk of Ferdi yesterday, and I'm going to go a little bit more into the detail because he gave a wonderful introduction already to the field. So we have two components in our energy system. So we can get renewable electricity from solar energy, from wind. We can use this for mobility. We can produce heat. But we can also work on using these green electrons, this renewable electricity, to split water and to make green hydrogen. We have Klaus saying today that he likes carbon. We have Ferdi saying that he prefers the hydrogen as his favorite element. I like them both because if you combine both, you can make a lot of things. Right? You can make platform molecules, you can make e-fuels, for example, green methanol, green ethanol. And you can do this taking combination of green hydrogen, for example, with CO2 that you can have generated from burning stuff or also from the exhaust of industrial activity. If you combine the green hydrogen with nitrogen, you can make also ammonia, and you can use ammonia directly for fertilizers, or as it was mentioned yesterday, it could be a hydrogen carrier. So we have electrocatalysis, which I will be talking about today. This interconnects a physics-based energy system with the chemistry and materials-based energy system. And we have the thermal catalysis, that is the basic of our current energy conversion processes in the world that gives us the e-fuels and green materials. So what do we need to do this? We talk a lot about making green hydrogen, but so far this has been a black box. So we need this, we need catalysts. And the catalysts are very complex. And the problem that we have is that no matter what we synthesize, it's going to change under reaction conditions. So imagine if this happens for an inorganic material. I cannot only imagine what happens with a biological material put on an electrode, how much this is going to change, and how much this is going to degrade, possibly. So what is a catalyst? The original definition by Berzelius was that the catalyst is a substance that can promote a chemical reaction without being consumed nor replenished. This sounds ideal, but we know by now that this is not true. The catalyst is changing, you get dissolution, you lose some of the material, it's changing in structure, it's changing in surface composition. And if you look at this picture here, this illustrates why do we need a catalyst, right? So a catalyst will change the velocity of a chemical reaction. It does it by modifying the energy profile of this reaction. So if you want to go from here to here, you can go all the way on top of the mountain. This is without the catalyst, the activation energy that you will need, or you can go through a different path. This is what you will have with a catalyst. But how do you go from here to here? So anything, the design of your catalyst and the changes of this material will influence these two energy profiles and the difference between the two of them. So we say that it's great to have hydrogen, we can get it from water, but this is not new, right? Think about technology transfer. When I'm talking to politicians, you can tell, it took us quite a bit. This was in a book from Jules Verne, and by that time already, electrolysis and water splitting was already discovered. And he wrote, water will be the carbon of the future, 1970. Tomorrow's energy is water that must be split, dissociated with electricity. The water elements generated in this way, hydrogen and oxygen, will ensure the Earth's energy supply in the future. So 1870, and we are still trying to do it. And We have made great advances, but we could have been faster. And why didn't we do this before? Hydrogen doesn't have a large energy density. We had oil, we have carbon, so these are energy sources with high energy density. And until we started to think about climate change and the problems of the planet, we didn't think about maybe we should be looking for alternatives, even if they might be more expensive, which in fact at the moment they are more expensive than other energy sources. So just to give you an idea, these are the dimensions of a water electrolyzer, a relatively small one. So this is a very large system. This is only for 3 megawatts. And this is an alkali electrolyzer. Again, to get an idea what we are talking about, you need 10 tons of water, 50 megawatts per hour green electricity, one of these electrolyzers with this hidden catalyst that I mentioned before that we'll be talking more during my talk today, and then you will make 1 ton of hydrogen. Ferdi already mentioned yesterday that it's very hard to transport hydrogen. If you make a pipeline, you need to make a huge pipeline if you want to transport it as gas. We know already that sometimes pipelines are attacked. Some people were talking about security, so it's not so ideal if you only depend on a pipeline. At the same time, it would be nice if you can transport it as a liquid and also using infrastructure that we have. We know very well how to deal with ammonia. So Fritz Haber already was dealing with the synthesis of ammonia in the 1900s. And so we can transform hydrogen combined with nitrogen into ammonia, and we can transport ammonia. As it was mentioned before, the only issue is that if you transport it as ammonia, you are going to have to either ideally use it directly as ammonia, or you will need to convert it back. And in this conversion, we will lose energy. So I mentioned before that since the introduction was already given, I will give you something that probably you have not seen before, and I'm going to be focused on the catalytic material. What do we need to do electrolysis? So we need a catalyst. And this catalyst is going to be, we synthesize something that we call the pre-catalyst. It's going to change during the reaction. You need to do an activation. There will be the surface structure. The composition is going to be changing. And if you're unlucky, you get the activation. This is all tuned by the local chemical potential. But the catalyst is not alone. There is an environment. You have your reactants here. We are going to have an electrolyte, for example. And then the reactants are also going one of these kinetic process. And so these two processes together, they are going to determine what happens to the material and how effective is this electrolyzer. Yesterday was discussed, yes, we have the technology, but how effective this is, how durable it is, and this is going to determine also whether we can implement it. So if you want to have a stable productivity, you need to have the active mass of this material morphologically stable, but you need to know that there is a dynamic surface that will be regenerating these active sites. This sounds easy, but it's not so simple. You synthesize your pre-catalyst, and then the catalyst in its active state is going to form during the chemical reaction. I put a video here because I think it illustrates very nicely. This is transmission electron microscopy, and it's going to show you how a catalyst is changing. This is a catalyst that we can use to make green methanol. You combine CO2 and green hydrogen, and you can make methanol. Here, these are nickel particles. The rest is a copper surface. And you see these things moving here, right? This is bad, because we only have some temperature, a very small amount of oxygen, and you see that these are atomic steps, and these atomic steps are already changing with this minute amount of oxygen at a certain temperature. So for many years, people in catalysis have been trying to explain catalysts are stable materials, the catalysts don't change under the reaction, and certainly they are not dynamic, and you can see from these pictures that this is not true. So there is still a lot of work to do. If we are thinking about how do we do this, so we spend a lot of time in organic chemists preparing a pre-catalyst. Then you go under reaction conditions. We don't know what is happening here. And then you get maybe a lot of details about the catalyst after the reaction. But if you want understanding, and the understanding will allow you to tune the characteristics of the pre-catalyst so that you get the ideal working catalyst, then you need to combine microscopy and spectroscopy under realistic conditions, and this means under operando conditions. So you are going to see in my talk how do we do this and why this is relevant for green hydrogen production. So this is just an example of how we make solar fuels. This is the conversion of carbon dioxide into fuels, for example, ethanol. And I just put this as an example, again, about the dynamics of this catalyst. We have CO2, we have an electrolyte that is potassium bicarbonate. I'm looking at all of this imaging in an electrochemical cell in a transmission electron microscope. And you saw that at the beginning, I make these cubic structures. These are these copper oxide cubes. But very quickly during the reaction, you see the mobility of this structure, dendrites are forming. So again, high complexity, and this structure and changes in the compositions are going to determine the performance, in this case, the activity and the selectivity of these materials. So going back to water electrolysis. So we have a relatively simple system. So you have here our electrolyzer. In the cathode, we are making the hydrogen with the hydrogen evolution reaction that is shown here. In the anode, we are making the oxygen with this reaction. And the hardest thing to do is to make the oxygen evolution reaction. And this is assigned to the difficulty of this sluggish kinetics at the anode for electron oxidation of oxygen. This requires an overpotential, and we are always working to try to modify this overpotential. But we know by now what to do and what are the materials that are working. So there are a lot of theoretical and experimental work, and we know that there are certain mixes of oxides, iridium oxide, and mixes of nickel, iron, and cobalt that are going to be very good catalysts for this. Ferdi mentioned that there are two technologies, one in acidic and one in alkali. And the problem is that one that is very efficient, the acidic, works with iridium oxide, ruthenium oxide. Just to give you an idea, 152,000 euros per kilogram. And you don't have iridium everywhere. So if you want to do a global energy conversion and you want to use PEM fuel cells, then I think you are in trouble, because we don't have enough iridium. It's extremely expensive, and it's not everywhere. The other technology that is now becoming much better is this alkali technology, and this works with materials that are much cheaper. For example, cobalt, 33 euros per kilo, a mix of nickel, cobalt, iron. And this, I think, is much more doable, and this is something that we are working now in the industrial implementation, in what companies also in Germany are very heavy working on. So what can we do in research, right? We already know there is an electrolyzer and where there are some materials that work. What are still the things that we can do? So there have been a lot of studies, even for these very nice cobalt oxide or cobalt hydroxide catalysts. There have been a lot of studies trying to make different pre-catalysts with different structures, with different compositions. And unfortunately, what we realized is that there was not much difference in what was coming out, because under reaction, there is a structural unification. So you start with a cobalt oxide or hydroxide, you put it in your electrolyzer, you apply the potential, you do oxygen evolution reaction, and then you make a surface termination. In this case, this cobalt oxide hydroxide, this is a skein layer forming on the surface. And then if you remove the potential and you look at your material again, you get again a cobalt oxide. So for many years, we wasted a lot of time looking at the pre-catalyst, looking at the after-reaction catalyst, but none of them was representative of what was the active material. So there is still things that needs to be done. Then this requires operando spectroscopy and microscopy. If you are thinking about what are the things that we can look at, we need to make the material more efficient, and we need to make the material more durable. And to do that, you can think, for example, about designing the pre-catalyst with different sizes. So we did this. We were looking at different cobalt oxide and cobalt hydroxide catalysts with different particle sizes. And when you decrease the particle size and you are below a certain regime, like 5 nanometers, you see that it's more active. So we can now understand more why this is more active. And you can go to a synchrotron. This is, again, why do we need major instrumentation? You can go to a synchrotron, have high energetic X-rays, and look at this, for example, with a spectroscopy method. This is X-ray absorption spectroscopy. You have 1 nanometer particles, very small particles, and you can see from this data that the as-prepared material in black is very different from the under reaction conditions material. So this already tells you that you need these studies to understand what is happening during the reaction. And there is also a size effect. If you look at particles with different sizes and you look under reaction conditions, for example, the oxidation state of cobalt appears to be slightly different. We know now that it's not so simple, that there is a charge redistribution between the cobalt and the oxygen, and that we have the formation of these oxyl species. This happens for these cobalt oxides. This happens also for iridium oxide. But this type of information is key in order to understand what material do I need to make my electrolyzer more efficient and also more stable, and minimize the amount of metal that I'm using, especially for the people that were working with materials like the iridium oxide. So when you are talking to the industry and you give them this wonderful data under ultra pure conditions, experimental data, they will tell you, yeah, but our electrolyte is not ultra pure. And what is the most common impurity that you have in water is iron. So we try to understand what does iron do to this catalyst during water splitting. So we make a catalyst, in this case, these were cobalt oxide particles, and then we put it in an electrolyte that contains iron. And just a small, very small amount of impurities. And you see that you start making these structures, so you get iron incorporated into your material. And at the beginning, if you look at the performance, this is here the current density, our activity, and you go from no iron in the electrolyte to iron in the electrolyte, you see that the catalyst becomes better until you get like a stable state. So if you think about this, and first of all, you need to see is the iron doing something, and we see actually that the iron is doing some redox cycle here, so the iron is part of the active species. And we can see also that under OER conditions, oxygen evolution reaction conditions, the iron oxidation state is also changing. So that means that it's not just an spectator, the iron is doing something in our system. And then the question is, okay, does it matter if I have a lot of iron impurities? Can I have too much? Can I start having poisoning? So this you can investigate with a combination, again, of spectroscopy and microscopy. So we look at this nickel oxide catalyst. This is one of the typical catalysts also for the oxygen evolution reaction. And we started adding iron, and we could make the catalyst better, but at some point we put too much iron, and you see that the catalyst starts to become worse. And though we can understand now why this is, so during the oxygen evolution reaction, we can see that at the beginning, in yellow, we see the iron everywhere on the surface of this electrocatalyst. After OER, if you continue running this reaction for a long time, you start seeing the formation of these clusters on the surface. So there is at the beginning, the impurities at the beginning are coming into your active surface, and it makes from this nickel oxide, one of these structures, a layer double oxyhydroxide layer, just from the impurities in the electrolyte. This is good, this gives you the activation. But if you keep trying to put iron, then you start seeing that the iron doesn't go more into the bulk of this material, but it starts accumulating on the surface as iron oxides, then your catalyst is dead. So talking to the industry and being able to do this operando spectrum microscopy is really important to try to understand why do they see degradation in these type of systems. So I wanted to show you just another example, what is another role that these impurities can have. The impurities can also affect the dissolution of the active material. I told you these catalysts are not ideal, and some of the problems that we have with these catalysts is that they are changing. If you look at these things here, it's hard to see, but imagine what I'm showing you here. So this is an cobalt oxide, in this case, platelets of cobalt hydroxide. This is cobalt hydroxide, and again, we are in a transmission electron microscope. We are under electrochemical conditions, and you see these small things forming here, these small particles. So this comes from the dissolution of our active catalyst. And the dissolution of this active catalyst is affected by the presence of impurities. So if you have the iron-free electrolyte, you see more of these particles that are dissolved and redeposit. And if you have the iron impurities, it's more stable. You still see some dissolution, but you see less of the dissolution. Again, a little bit of iron is good. Too much iron starts to create problems. Thank you. So, thank you. To be able to get more understanding, some of these systems are very complex. I mentioned before we are working with inorganic materials, but when you are working with a nanoparticle, you need to consider the effect of the particle size, the shape, and the changes of these structures, the surface composition. So sometimes it's good to start with thin films, because if you make these thin films, you can control, for example, the surface termination. And we can make these thin films with different orientations. You can also control the initial composition. But remember, this is just the pre-catalyst. But if I know very well how the pre-catalyst is, in this material changes under reaction conditions, I will be able to find out what are the important parameters to consider when I design my material, my catalyst for hydrogen production. And so we do this, and I want to show you just an example. This is, again, oxygen evolution reaction. We're making green hydrogen. For this reaction, you are making, this is the part that is making oxygen, but this is in a water splitting catalyst. And you have two different oxides. only the surface orientation is changing, same film thickness, same material composition. And when you look at the electrocatalytic performance, it's different. This particular surface orientation works better than this one. Okay? This is very useful to know, and then we want to understand why is this, and we can look with atomic force microscopy at this material under potential control in the electrolyte, how it's changing. And you can see that this roughness, these little things that are forming here, this is this active surface oxyhydroxide, this cobalt oxyhydroxide that is forming there. So we can follow this under reaction conditions. And if you look at this particular surface, same material, just different surface orientation, it's much more stable. There is still an increase in roughness, but it certainly is changing in a different way. And when I have this data, then I can go back and say, I'm going to try to design my electrocatalytic material, my nanoparticle, maybe with more preferential orientation here, even though we know that it's going to have certain modifications during the reaction. So the other thing was that since I know that the iron impurities are doing something, I can try to put the iron already in the exact amount in the precatalyst. So when I do this, this is, for example, a cobalt iron oxide system that I made, a thin film, just the same orientation. And this is as prepared material. These are scanning tunnel microscopy images. These are atoms. You see defects, so these thin films are not perfect, but this is the reality is like this. Nature is not perfect either. So after the reaction, you can see that the material change. You see more roughness. In these images that are low energy electron diffraction images, you see already that something happens. So this disorder of the surface indicates that we have the formation of this thin oxyhydroxide layer. And by now we can measure and we know that this is less than 1 nanometer. So when we are looking at these systems, we can look at our initial material and compare pure iron oxide, pure cobalt oxide, and this cobalt iron oxide. And at the beginning, the cobalt oxide was better than the other two. But if you run the reaction for a couple of times, because of these changes that I mentioned before, the catalyst is dynamic, then it turns out that the best catalyst tends to be lower overpotential, tends to be the one that includes a little bit of iron. And within these thin films, we can be very quantitative. So we can look at how much oxygen we are producing, and we can look under reaction conditions, and we can look at the beginning. And again, at the beginning, we have the cobalt oxide more active than the cobalt iron oxide and the iron oxide. But under the reaction, after the material has changed and became in the active state, then the cobalt iron oxide is the most active working catalyst. It has the most active oxyhydroxide. What we learn also with this detailed characterization is that the thickness of the active component of this catalyst, this oxyhydroxide layer, depends on how much iron do I add. And if you have a lot of iron, I have a thinner oxyhydroxide layer. So these are the things that I wanted to tell you about today. So I will give you some take-home messages. So I hope that from my talk you could see that the operando spectral microscopy studies are really needed to understand the electrocatalysis, or in general the field of catalysis, to get basically knowledge-based rational design of your material. The material that you make is a pre-catalyst, so we can spend a lot of time doing the inorganic synthesis. This is very valuable, but we need to understand that this is only the precursor and that this material is metastable and this can adapt to the reaction environment. So the structure will affect the adaptability and the subsequent operando transformation. So yes, spend time making your initial material, but also spend time characterizing it under reaction conditions. And because the active state of the material is only formed while the material is operating. The active sites are dynamic. I hope that I could show you this even with videos. And if you want to get mechanistic understanding, then you need to really be looking at the material while it's performing the catalysis. With the example of iron, I try to illustrate that very small amounts of metal impurities on these metal oxides, they tune the local structure of the active sites in morphology and electronic structure, and also that the dynamics of the active sites must be coupled with the kinetics of the reaction. I want to finish with a sentence from Albert Einstein, because I think this describes very well the work that we are doing. Everything should be made as simple as possible, but not simpler. So a catalyst is a very complex material, and the catalyst changes. And so we need this complexity, and we need to be able to understand such complexity. I just came back from from the birthplace of Rutherford from New Zealand, and he had a very nice quotation in his museum: "No scientist, however brilliant, operates in vacuum. The process of groundbreaking discovery only happens in a collegiate context that spans many generations of scientists." This is my research group, and I'm here representing the work that we are doing, but all the credit goes to these people. And I will be very happy to use my entrance in the academy here to also set up new connections with people in the audience. Thank you.
Thank you for a very good talk, and welcome to the Academy. So you have a brutal number of influence factors. How far is theory helping in your design? And the other thing is, the catalyst would work in a reactor. So how much does the catalyst design and the reactor design influence each other?
Okay, to the first question, we are working very closely with theorists, and I think we need to be, especially when you're working in electrocatalysis, you need to be very realistic of what can a theorist calculate at the moment. So you need to have your material, you need to have the applied potential, you need to have the electrolyte, you need to have cations from the electrolyte that might even become intercalated in some of these materials. So all of this complexity cannot be addressed at present by any theorist. However, if we have, for example, experimental data where I can tell the theorists, don't start with the pre-catalyst and try to look at the transformation, because this is something that is very hard to do. I'm telling you already, this is the structure that has formed under reaction conditions. Now help me to understand, for example, when I do some spectroscopy, a vibrational spectroscopy, I see some signatures there. Help me to understand what are the species intermediates that could correspond to these intermediates. So the restructuring is also being looked at in theory, but I think you need to divide the problem one step by one. When we're looking at these thin films, or single crystals, you can work very nicely with theories, because I can tell that my active catalyst has this surface termination. At least they know which exact composition I start with and the surface termination. But if I tell the theorist, calculate how much leaching I'm going to get from this material with this perfect initial surface composition in an electrolyte, that gets much harder. The second part of the question is how much is the reactor, how much is the catalyst? The catalyst is what I illustrated here. Of course, the reactor also matters. And I think you cannot consider one without the other, because in the laboratory, we are working at very small scale. So we always have collaboration with industry, because then they can test the materials under much harsher conditions. The first is that we work under much cleaner conditions than in the industry. The second one is that in many of these experiments, we are working at low current density. If you go to much higher current densities, the degradation happens much faster. But we can do this. We can try to take some of the industry samples, investigating ourselves, and we can also try to go under more realistic conditions. But I think it's very important to try to collaborate, because you don't want to do all of this detailed characterization under irrelevant reaction conditions in an irrelevant reactor. So,
yeah, Brett, very interesting talk about the really fundamental understanding. I just wonder, have you ever tried to integrate your OER catalyst and with the HER, the cathode in the real electrolyte condition? How about the onset potential? Is it get much decreased compared to the state of our catalyst, or how it looks like?
Depends on the system. So we have not been trying to get record low onset potential. We have been trying to understand what is the origin of the changes. And so in many cases also people are reporting these record onset potentials under conditions that are completely irrelevant to any industrial application, or they are reporting it for very short periods of time. And so you want to see how this changes. So yes, we have this type of measurements. My focus has not been on the optimization of the onset potential, more on the understanding of what do I need to do in the material, in the reaction conditions, to get more stability and also to get more activity.
So you mentioned faceting and surface termination, and so faceting. Do you know which are the facets that might be most effective?
You saw the example here for this cobalt oxide species, this 100 seems to be much better. Again, you see changes, but what they do this on the 100 surface, it looks like the structure of the highly disordered oxyhydroxide that forms seems to be more active. For other reactions, for example, for CO2 electroreduction, we know that there are different roughness that you need to consider. You have the roughness at atomic scale, and you have the mesoscale roughness. And we know by now that under reaction conditions, you are making steps on the surface, in that not only the step density matters, but also the step orientation. This is why we went back to looking at these thin films, to looking at single crystals also, to try to address this. Yes, we can look at these things for the OER, for example, 001 surface will be better, but for every reaction, you need to look at this, and again, look at over many hours and see how the surface has been changing, and then you see the step orientation is changing. So once I talk to my theorist colleagues, I already tell them, you know, I run this reaction for X number of hours. These are the steps, kink steps, that are the most active. Then they can do the theory, and they can try to explain us what is happening on these kink steps.
Sorry, how much do you know about the influence of the spin texture that this type of surfaces of this cobalt oxide, hydroxide have on the mechanism?
So this will be, to tell it in a different way, you worry about the structure of, in this case, we have epitaxial films. In my example, these were highly ordered, but then the surface becomes very disordered. And what we know is that there is some leaching of some of the components, for example, this iron out of the cobalt oxide, and this depends on the structure of the underlayer and on the thickness of the overlayer that forms. But these are things that we can measure, so this is, again, system dependent. It depends on where you grow these materials on, and yes? The spin structure.
And how the electrons are paired of the cobalt.
There is a lot of discussion in water splitting electrocatalysts of the possibility of having an effect from magnetic fields. And there are some papers published recently claiming that this is the case. And they have seen some effects, but honestly, there is no explanation still whether this is really related to the spin configuration. I don't believe the explanations that were published until now, I don't think they are sound. They see an effect, but whether this effect is due because of reorientation, even structural reorientation in the presence of a magnetic field, there are people that do electrocatalysis, they do electrogrowth, so they do growth of materials in the presence of a magnetic field. And this, for example, changes the orientation. So in the data that they have published, I cannot tell you whether there is really a spin effect or if it's just another effect of the magnetic field, including these changes in the structure, maybe.
Bingo. Maybe it was this, because the structures of cathode active materials are very related to this, and leaching there from depends on the oxidation state, because then for the spin again back you get Jahn-Teller distortion, and especially if you have Jahn-Teller ions which are distorted, they easily leach out, and so one thing is the conservation of the oxidation state at a level where you don't get any Jahn-Teller distortion. So have you seen such things?
So again, I have not measured the magnetic properties of these materials under reaction conditions. We need to be very, very careful to say you can characterize the pre-catalyst, you can characterize the final catalyst, but under reaction conditions, you need to do all of this characterization. And what we have seen is, yes, if you have under reaction conditions, different oxidation states, this is going to affect the performance. And the leaching, yes, it will depend on this oxidation state. But then it will be, is this a direct effect or is this an indirect effect that maybe you have a modification of the oxidation state and maybe what is happening is the leaching. So that's why I say, I believe that people have seen some effect. We tried to reproduce one of the Nature articles that was published, and with a very small magnetic field, in a material that was, my PhD was in measurable spectroscopy, so I have a background in magnetism. And we tried to reproduce one of these experiments, and to tell you the truth, we couldn't get this effect that was reproduced. So I'm extremely careful there. I'm not saying that this is impossible, but I'm saying that there's a lot of much more research that is needed. Because at the moment, you think about the magnetic materials that people are reporting, and you think that about using not an electromagnet, a permanent magnet with a very, very small magnetic field, then you need to see how much, if this will be really sufficient to change anything. And my own experiment test of this paper was not reproduced.
Well, I think this tells us there's a lot of work that's left to be done. That's not going to happen now, so please join me in thanking Beatriz and all the speakers of the session.